Meiosis is a process where a single cell divides twice to produce four genetically unique daughter cells. This is a form of sexual reproduction. The four daughter cells contain half of the amount of genetic information found in the parent cell. This means that at the end, one diploid cell becomes four haploid cells. The cells at the end of meiosis are called gametes, or germ cells, which are also our sex cells. There are 8 stages in meiosis: prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. Each cell has a lifespan, or a cell cycle. During the cell cycle, a cell grows, prepares for division, and then divides. The cell cycle is split up into 4 phases: G1, S, G2, and M. M stands for mitosis or meiosis. Interphase, which is what the G1, S, and G2 stages fall under, is a phase of the cell cycle that the cell stays in for the longest time. The G1 and G2 phases are periods of growth and intense activity.
Interphase, G1 phase, cell growth. G1 is the first gap or growth period. The cell does most of its growing in the G1 phase. The cell increases in size and synthesizes new proteins. S phase, DNA replication. New DNA is synthesized when the chromosomes are duplicated. At the end of S phase, the cell contains twice as much DNA as it did originally. The DNA is duplicated in order to divide. G2, preparing for cell division. In G2 phase, the cell must grow some more and produce any molecules still needed to divide. This includes the assembling of microtubules needed to create spindles during mitosis. The cell also must duplicate its organelles to make sure each daughter cell gets everything it needs. At the end of G2, the cell must make sure all of the proper structures are present and ready for cell division.
Prophase 1. During prophase 1 of meiosis, the chromosomes in the cell condense and become visible. During this phase is also when crossing over and synapses occur. Synapsis is when the homologous chromosomes are paired. When they have been paired up, the strands of homologous chromosomes cross over. The chromosomes exchange segments with each other. This process is known as genetic recombination. By doing this, it results in genetic variation among the gametes. Also during prophase 1, the meiotic spindle forms and the nucleolus and nuclear envelope disappear. Metaphase 1. During metaphase 1 of meiosis, the pairs of homologous chromosomes align along the metaphase plate, which is along the center of the cell. The centrioles move to the opposite poles of the cell, and the spindle fibers attached to the centrioles coordinate the movement of the chromosomes. This helps ensure that the chromosomes separate properly. The spindle fibers are attached to the centromeres of each homologous chromosome.
After metaphase 1 is finished, the cell enters anaphase 1. During anaphase 1, the cell starts to expand. The spindle fibers pull each homologous chromosome toward opposite ends of the cell. Instead of breaking apart like in mitosis, the sister chromatids stay together. Once anaphase 1 is completed, the cell goes into telophase 1. Telophase 1 is the last phase of meiosis 1. In telophase 1, the separated chromosomes cluster at the opposite ends of the cell. During this phase, a nuclear membrane forms around each cluster of chromosomes. Cytokinesis, which is when the membrane creates a cleavage furrow and splits into 2 daughter cells, follows telophase. By the end of telophase 1, there are 2 haploid daughter cells formed.
Prophase 2. Prophase 2 is the first stage of meiosis 2. Now there are 2 separate daughter cells going through these stages. Unlike in prophase 1, there is no duplication of chromosomes during this phase. The centrioles duplicate just like in meiosis 1, as well as how the spindle apparatus forms. Also, there is no crossing over in prophase 2 because there are no longer homologous chromosomes being lined up, but rather just the sister chromatids. Metaphase 2. After prophase 2, metaphase 2 is the second stage of meiosis 2. During this phase, single chromosomes line up along the equator of the cell. Similar to mitosis, there is only one singular chromosome instead of two homologous chromosomes like in metaphase 1. The centrioles have moved to the opposite poles of the cell, and each daughter cell has formed a spindle apparatus.
Anaphase 2 is very similar to anaphase 1. During anaphase 2, the cell begins to expand. The sister chromatids break apart, and the spindle fibers begin to pull them to opposite ends of the cell.
Telophase 2. Telophase 2 is the fourth stage of meiosis. Cytokinesis takes place. Chromosomes reach opposite poles. The two cells produced by meiosis once split. This forms four haploid daughter cells and nuclear envelopes. Once telophase 2 is over, the nuclear membranes of the two cells are fully formed, completing meiosis altogether.
At the end of meiosis, four genetically unique haploid daughter cells are formed. Meiosis introduces genetic variation and new combinations of traits, providing infinite variations. It also maintains genetic similarity and differences.